And before I go over the slides, I would like to just remind you folks before I forget. Today we have 4 presentations, and also today we'll have our final review assignment, which is a bigger take-home quiz, basically a summary of what we have covered since the midterm, because there's no final exam. So we're going to use this quiz to review and assess your understanding of all the different conceptual issues. So this take-home quiz will be, there are 18 multiple choice and true or false questions in total. So which means, well, basically everyone will have 2 attempts, and I'm going to take your higher one. So basically it's not timed. And after you finish the first one, you'll be able to review each question and see the score out of the total. So that will give you an idea of whether you did this question right or wrong, so that during your second attempt, if necessary, you can try to focus on those questions. So this quiz will be released after class today, and you will, so I'll be posting a link here, or you can also go to the quiz page directly. And this is due by Wednesday next week. So you'll have about 10 days, 18 questions in total. So that's a schedule reminder. And today we're going to talk about vegetation indices. Some of you have read about ndvi, and this is what we're going to talk about today. And afterwards, we'll have 4 presentations on remote sensing applications, and there will be 7 more next Monday. So if you're not presenting today by the schedule, then you'll be presenting next Monday, not Wednesday. Wednesday, we're going to work on the last lab, lab 9, on ndvi in Earth Imagine. So I will try to keep my part short so that we can leave some transition time or discussion during the presentation. Everyone will have roughly 10 minutes just for the time limits we have. So I'm sure so far you should be very familiar with the typical spectral patterns. Quick question, what are we reading here? If I ask you to guess the land cover represented by this particular spectral signature plot, should be easy. So very high on near infrared portion, and during like over the visible portion, relatively high on the green band.
Some kind of vegetation.
Some kind of vegetation, sure. And that's something we have been talking about, and also a reason why we use the false color near infrared combination whenever you display images, whenever you have this large amount of vegetation or even in urban areas. So for the vegetation indices today, this is, well, basically this spectral signature plot is the foundation of how many of the vegetation indices were developed. And it's very typical for a healthy green vegetation, some trees and some grassland, and depending on the species or growing stage, the patterns can be slightly different. But overall, we see very highest near infrared reflectance and a little bit of peak on the green band during over the optical portion, the visible portion. I think we just briefly mentioned, but here I just want to give you the statement or this information on why this is the case. So I think it's easy to understand why green vegetation has a high reflectance over the green portion in the visible portion, because, well, we are talking about the color. So for a certain object with a certain colors, they definitely reflect higher on that particular color portion. So that's about the pigments. And also what determines the high reflectance on the near infrared band is actually the actual internal cell structure, the leaf structure, which leads to the high reflectance in the near infrared band. So which is to say, both pigments and leaf structure affect the spectral signature plot for some trees or some leaves, some grass area. So which means any like minor change in terms of the structure. For example, we talked about the moisture content. We talk about the seasonal change, which can alter the pigment, the color of the leaves or the internal structure of leaves, and those can all change the signature plot. So that is why, even if you plot for trees, the same species, you will still see some variations just because of this color difference and structural difference. And this is certainly the basis where we want to use or make use of when applying the vegetation indices. And here I want to bring in a concept called the red edge, which is part of the review question. So you will have to review either ways. So the red edge in the context of remote sensing or vegetation analysis, it refers to this transition, this fast transition between the red band to the near infrared band, where we see a sharp increase, a jump of spectral reflectance between the two portions, because we know the whole spectrum is continuous rather than discrete. So which is, which basically means there's like really no sharp cutoff. So the red edge refers to this transitional portion between red band and near infrared band whenever we're looking at vegetation. And this portion is important for mapping vegetation, also for defining vegetation indices. So the next slide is an example from the ArcGIS imagery book, and they're mapping ndvi, and they call this image red edge. And because ndvi is specifically making use of the difference between red band and the reflectance on red band and near infrared band for healthy vegetation. And in this particular case, and they're giving the higher ndvi values a color of green, and then the lower ones are on the purple. So, somewhere in the middle, they're basically the moderate ndvi. So higher ndvi, as we'll be introducing shortly, they refers to the very, high amount of vegetation or very vigorously growing vegetation. And this is just one example. And also just a quick idea. So once you calculated ndvi from your original image, your results will basically be a raster layer, and each cell in your raster layer will show the ndvi value. And in Lab 9, you folks will see how to calculate ndvi within ERDAS Imagine. So ERDAS Imagine, also just similar to many other remote sensing program, they have implemented many vegetation indices and also other image indices into the program. So which means calculating an ndvi is very easy in ERDAS. So basically you just choose ndvi and you plug in your image and you hit run and you get your results. But also, the reason why we introduced the spatial modeler in the last lab during the image differencing, so which will offer you an opportunity to calculate any image indices. For example, if you're reading an image index from some publications, which is not part of ERDAS, you can apply spatial modeler to calculate any indices on your own. And the same thing in ArcGIS, you can use raster calculator to calculate any indices on your own. So just the technical idea, and you folks can read more or learn more in Lab 9. So vegetation indices, and basically they are some equations, some mathematical equations, and usually very simple, and to try to measure the total amount or the growing growing status of your vegetation cover. So basically, after you compute some vegetation indices, every single area or every single pixel in your image will receive a index value, which represent either the amount or the relative amount or the relative vigorous of vegetation within a pixel. And also, I want to highlight the word relative, because what you compute from the vegetation indices will be a value as opposed to a category. So you wouldn't really know whether it's vegetation or not vegetation based on only the vegetation index. It will only give you a value whether it's 1 or whether it's 0.5. Depending on which index you measure, the range of values can also be different. So they will give you a relative sense of it's higher or it's lower, and it's up to the analyst to determine what the actual land covers are. And also you can definitely use the information maybe from the original image to as a reference, or field surveys can also give you additional information on what actual land covers are. So today we're going to focus on two basic vegetation indices. The first one is called the IR, which refers to near infrared, and R refers to red, the IR and R ratio. And the second one is our focus for today, and also what we will be talking about in Lab 9, the ndvi, Normalized Difference Vegetation Index. So just a quick review on what the patterns are, because we mentioned there are many factors which may contribute to the spectral signature, or the difference in terms of the spectral signature for a some sort of vegetation covers. We mentioned both the pigments and the structure can be important. So first of all, certainly seasonal change can play a big factor. So this is a composite, a mosaic image from Landsat, and to show the fall color near the Great Lakes area, and basically to show you the seasonal change. And if you're analyzing the fall season image, which is basically to say your green portion won't be really as high. So there are a lot of seasonal factors that may contribute to the variations in terms of spectral signature. And the second example is showing you how pollution or any other human activities may lead to difference in terms of spectral signature. So A here is a pollution source, industrial pollution source, and the two nearby forest areas, B areas, and you can see just only from the color, without any field observation, you can see the potential influence on these two patches of woodlands. And all this, either seasonal factors or external human factors that can all affect how your vegetation looks like on an image, and also affect how the results from your vegetation indices will reflect. So first of all, for the IR and R ratio, it's very simple. It's just a simple division. You divide the cell value from your infrared band by the cell value on the red band. And this will roughly represent essentially the amount of vegetation in a particular area, or even no vegetation. And also I want to clarify that we're talking about vegetation indices, but whenever you're calculating the vegetation indices, you're calculating for your entire image, which is to say your urban area, for example, the buildings, the stadium, or the farmland, or the water, the rivers, they all will receive some sort of vegetation index value, which will likely be depending on which index you use, and relatively say they're going to be lower. For example, in your water area, you will see a very low IR and R ratio, because we know the IR of water is almost 0. So you'll likely receive a near 0 index value for your water area if you use the IR and R ratio. But everywhere in your image will receive some sort of value, and use that value to represent the relative abundance of vegetation, or the growing status of your vegetation area. So in this case, we know the IR for vegetation is going to be higher. So apparently higher IR and R ratio will represent, relatively speaking, once again, actively growing plants. Oftentimes this information can be used in a relative sense, because, for example, if you're analyzing vegetation change over time, maybe after a some sort of disaster event, maybe following a flooding event, you can calculate the IR and IR ratio for multiple days following the floods, and then you can analyze how the vegetation change or visualize how the vegetation change following the disaster events, and to see the vegetation recovery or the how the event has impacted the local vegetation growing. So that's one way of using it. And alternatively, some people may use the vegetation indices more similar to a unsupervised classification. And actually in Artist Imagine, you will locate the ndvi tool under unsupervised classification, because it's very similar. You just run this process and the computer program will calculate ndvi for you. And afterwards, you can use original image and use Google Earth or use your field observation data to help you determine a threshold value for you to classify your image. Because typically vegetation will be relatively higher, and you can analyze the vegetation area versus non-vegetation area and try to determine a threshold value to classify your image into vegetation and non-vegetation. And that is another use. And in Lab 9, the one I'm going to release later today and for you to work on Wednesday, you will see, well, to save time, I actually gave you a threshold value to help you classify. But actually, it's going to be a long process, very similar to how you label your different clusters through unsupervised classification. You probably have to, dynamically link your ndvi or your other indices image with your original image with Google Earth and trying to determine a threshold value. So that's a simple IR and R ratio. So the problem with the IR and R ratio, as you folks can imagine, we know the red edge, we know the sharp transition from red to near infrared, which is to say, the value will get really high whenever we're looking at vegetation. So which is okay. However, whenever we're talking about calculating some metrics or some indices, usually we want to normalize so that we do have a general reference on what is high means. Because in this case, IR and R ratio can get extremely high. Maybe you calculate an IR and R ratio that is higher than 1,000, which is too high to have any relative meaning. So to solve this problem, now ndvi is more commonly used. So ndvi refers to a normalized difference vegetation index, and usually you can just go with ndvi. However, if you're writing a paper, the first time you mention ndvi, you want to spell it out. So ndvi, so this is the formula. So you first of all do a subtraction between your IR and R, so your infrared and red band, and then that is divided by the sum of the two. So for based on this normalization, your ndvi value will be constrained to a range of -1 to -1 to +1. So make sure that, just have the sense that ndvi, because again, you're calculating ndvi for every single pixel, every single cell in your image. Your water area, once again, if we do this subtraction divided by the addition, your water area will definitely have a negative value. So that's really why your ndvi will have a range of value that is ranging from -1 to +1. But this will give you a relative sense on, certain areas are low, certain areas are high, but the range is not crazy. You still have, this limit from -1 to 1. And definitely, once again, your healthy vegetation will show a relatively higher ndvi value. And there's no specific threshold, to say how high is vegetation. So once again, it's kind of like a unsupervised classification is, it really depends on your study area, the season, the particular context. So usually you have to determine, if you need a threshold value, you have to determine the threshold value based on this particular image by, looking at the source and, looking at all the information together. But there's a well-documented problem, and this is something you will need to keep in mind because we do have a question in the review quiz, but and also in the meantime, that can save you a lot of effort if you're trying to apply ndvi. So the fact is, if you have done enough literature review of ndvi, or if you can just do a quick search on Google Scholar to do like ndvi, like issues or challenges, you will see a lot of publications talking about this saturation problem of ndvi. So which is to say, let me just try to translate this a little bit. So some people try to use ndvi to analyze the seasonal, well, basically the change in the dense vegetation area, for example, the tropical rainforest. So what they found out is, as you folks can imagine, we're not really talking about the deforestation, that sharp of change. It's just more of the relative abundance in the tropical rainforest, the natural forest area. What they found out is, even if the actual abundance or vigorous can be different on the ground. However, ndvi tends to be very similar over the rainforest regions. And also, if you can imagine a diagram, so what this means is ndvi functions well when the relative abundance of vegetation is low. So you can expect a linear line, whether it's flatter or it's steeper, as we look at the relatively lower vegetation areas. However, when we reach to like where you see over 80% or 90% of vegetation, the ndvi doesn't really distinguish between the dense vegetation areas. So this is the problem. So you see, like, if you can imagine the curve will flatten as they grow up to the higher density vegetation areas. So ndvi, in that case, is not very useful for tropical rainforest. This like observing the relative amount of vegetation. And also, just in the meantime, because today we're going to focus, we focus on ndvi. There are many new vegetation indices being developed, and there's a newer one called Enhanced Vegetation Index, which is used to solve the saturation problem of ndvi. And some research are still being done to develop other types of indices. And today we focus on vegetation, but also there are indices that is used to capture urban, for example, and some indices and used for some geological applications. And all of this are possible. So once again, the reason why we introduced spatial modeler last time was because, that way you can use spatial modeler to calculate any indices, even if you develop a new index or you're experimenting a couple index to really to show which one can be used to better capture a particular type of vegetation or maybe a particular type of land cover. So so that will be an option if you can use the spatial modeler to calculate other indices that is not implemented in Earth's Imagine. I think I'm going to actually stop here because I do have a couple more slides on possible applications of remote sensing, which I think we can prioritize you folks' presentation. And then if time allows today, I'll go over some of the other applications, and maybe you already covered, or if it's something new, I can also briefly mention, or I can just save the information in the slides for you to review afterwards if you're interested. So basically the next couple of slides, there are so first one, I had a video on the use of image analysis in archaeology. So actually it's one of the major application, as we showed how LiDAR was used to discover this huge scale of Mayan settlements back then. So which is a very commonly used application. There are also a couple other like geological applications and also social applications. So I'm going to just stop here, and I will stop